Hello students, my name is Neha Bendre, Assistant Professor in Zoology at Rare Shikshan Samstha's RBNB College, Sri Rampur. So today I am going to discuss you with the topic that is called as cytoskeleton. Okay, so this topic belongs to ZY336, that is paper number 6 for the BSc 3rd students. Okay, so this is the paper called as cell biology. In cell biology, we are going to study a unit called as cytoskeleton. So if we talk about the cells are a very minute bodies, okay, that is present in your system. The cell is the thing that forms the body, that forms the organism, that gives life to the organism. So what is the cell? The cell is the structural and functional unit of life on which the whole life depends upon. Okay, so in the cell we are going to study a very small part and very interesting part that is called as cytoskeleton. So students, I would like to tell you something about the skeleton that is already present in us also. Okay, so the same kind of skeleton is present in the cells too, though it is not seen with our naked eyes, because all the cells are very microscopic, which cannot be observed under which can be observed under the electron microscope. Okay, they are very nanometers in size. So here, as we have skeleton in our body to support our organ system, to support our to give rigidity to our body, the function is given by the skeleton. Okay, in the same way, the cell also possesses a certain kind of minute skeleton that is called as cytoskeleton. What is the term called as cytoskeleton? The cyto means the cell and skeleton means the dried body present inside the cell. As we have a cartilaginous body made up bony body, okay, just same as we have different parts of that skeleton, that is we have a cranium, we have limbs, we have a vertebral column, we have forelimbs, hind limbs and bones supporting that limbs. Okay, in the same way we have for the cytoskeleton, we also have some different kind of structures that makes up the cytoskeleton. Okay, so before starting with the different types of cytoskeleton, I would like to define the cytoskeleton. Okay, so this cytoskeleton are nothing but the interconnected filamentous tube-like structures. Okay, interconnected, which connects different kind of filaments. Tube-like structure, how are the skeleton? That is forming the tube-like structures, okay, that gives or that supports the cell shape, that maintains the structural integrity of the cell, that makes the cell move from one place to another. Okay, that is called as, it maintains the motility of the cell as well as it helps in cell division. Okay, so these many properties are maintained by the cytoskeleton. So it is a very important part of the cell structure. Okay, so in this, as we have gone with the definition, now we are going to see about different types of cytoskeleton. So cytoskeleton is made up of 3 types. Okay, so what are the 3 types? These are the microtubules, intermediate filaments and microfilaments. So as you can see in the words, it is formed, the cytoskeleton is formed of the tubules, filaments, okay, which are very micro in size. Okay, these three types we will study it later on deeply. So now let us see about the functions of cytoskeleton. What all properties are maintained by the cytoskeleton? So as I have said you earlier, it maintains the shape of the cell. If there were no cytoskeleton in the body, the cellular shape won't be circular or any kind of the cell, because in bacteria also we see that bacteria are also circular in shape, some are rod shaped. Okay, so that shape is given by the cytoskeleton. Okay, though they are not able to see with our naked eyes. Okay, so that circular shape is given through the interconnected mesh like filaments. Also, it maintains the structural integrity. So what do you mean by structural integrity? Whatever structure is present inside the cell, so it anchors, that organelles are anchored to that structural filaments. Okay, so in the cell there are different kind of organelles present, that is called as, here you can see this is a cell, structure of cell. Here you can see there are different kind of organelles present in the cell structure. So you can see there is a nucleus, there is endoplasmic reticulum attached to it, mitochondria, Golgi bodies, peroxisome, lysosome. Here one thing you can see, that I am talking about the cytoskeleton, that is giving a structural integrity. So all these cell organelles are supported by the filaments. Here you can see in the pink color, these are the filaments that are holding the cell organelles together. So that is giving an anchorage to all these cell organelles. So the pink color, filamentous mesh, which is forming a mesh, which is forming a net where all these organelles are maintained at their position. So if you see about the location, if you talk about the location of the cytoskeleton, so where are they present? They are present beneath the plasma membrane. So if this is a cell membrane or a plasma membrane, absolutely beneath the plasma membrane, the cytoskeletal structure, that is filamentous structure, is present, which maintains the cell shape, also maintains the cell organelles at their position. Okay. Also along with this you can see that here I have written, it helps in the formation of some bodies called as cilia, flagella and centriole. So here you can see some animals or some cells have cilia or flagella for the cellular movement. So that movement is given with the help of the cytoskeleton. Okay, which kind of this type is helping in the movement, I will tell you later on. So this, here you can see in the formation of centriole also. In this, here you can see the centriole. This centriole is formed with the help of the cytoskeleton. Okay, so what is the use? Why? Because centriole is formed during the cell division, to divide the cell. That time, at the time of spindle formation through the centrioles, which helps in the division of the cell, that time this centriole is formed with the help of some cytoskeleton elements. Okay. So this is clear that cilia, flagella and centriole are formed by the cytoskeleton. Also I have told you that to make the cell motile, or to give the movement to that cell, who is helping? The cytoskeleton elements are helping to carry out the cell from one place to another. Also there is one more important factor about cytoskeleton. Okay, one element from this microtubules, intermediate filaments and microfilaments, which is also called as actin. Okay, these some proteins like actin, myosin, tropomyosin are present in this element. So that helps in the muscle contraction. Whatever contraction is taking place in the muscles, this is due to that filaments present in our muscles. Okay, so this was all about the definition, the location, the functions, the structure of cytoskeletons, and also along with the types. So now further we are going to see one by one about different types of cytoskeleton structure. Okay. So next we are going to move on with the types of cytoskeleton. So in these types, first we are going to see the microtubules. So let's see what are the microtubules, and let's see the structure, the biochemical composition, okay, and overall assembly of microtubules. Okay, so first I would like to separate the term micro with small in size, tubules with the pipe-like structure or tube-like structure. So the word is microtubule, that is small tube-like structure present in the cytoskeleton. Okay, so here let us see which are how is the structure. So structure is hollow tube-like structure. So very importantly, hollow tube-like structure. So these why in the cytoskeleton, why there is a tube-like structure? The tube-like structure is due to, the hollow structure is to pass or to transport some ions or substances from one place to another, to carry these ions from one organelle to another. So this microtubules helps in the cellular transport. Okay, so there is a hollowness present in that tube, and so it is a hollow tube-like structure. It is made up of protofilaments. So this microtubule is made up of some proteins, okay, that is called as tubulins, which then forms the protofilaments. Okay, we will look out in the next few minutes. Now let's look at the function, that why microtubules are present in the cell. What is the reason? Okay, just as we know that in the regular building structure, okay, here also as we have in our body, there are different kind of tube-like structures, means the blood circulatory system. Okay, what it does, that is the pipeline of our body. It carries blood from one place to another. Okay, in the same way, the cell also requires some kind of transport in the cell, the liquid transport. Okay, so there microtubules are present for carrying out these substances. Also, if you see in the building structure, there are some, how is the skeleton of that building? So there are some rods and bars which make up the structure of the building, and also in that building only, with the help of that rods and bars, some pipelines are carried out to connect the water system in the building. In the same way, here also we are having these microtubules to carry out the cellular transport, to give the shape, okay, to carry out cell motility. Okay, so in this we will see the first, that how it is maintaining the structure. Okay, with the help of hollow structure. Here I have drawn the structure of microtubules, showing some tubulin or some ball-like structure. Okay, so I will come to the electron. So this is clear that it maintains the structure of the cell. Next is it helps in the movement of the cell. Okay, that is the motility. It maintains the cellular motility. Next is it is helping in the cell division. At the time of cell division, there is a cell spindle formation. Okay, that is coming out through centrioles. So the first thing is that centrioles are formed with the help of the cytoskeleton, and which is that element. So microtubules are helping in that formation, and in that spindle formation are formed basically by microtubules. Okay, at that time we can see during the cell division there is spindle length is elongated as well as it is shorter. So who is helping that? That process is done by the microtubules, where the length of that filament is made long as well as it is made short. So you will see how that process occurs. That is nothing but the microtubule formation. Okay, so it also helps in cell division. Okay, now after knowing the functions of cell structure, maintenance, cell motility and cell division, we will look at the structure of the microtubules. Here you can see a tube-like structure. Here in this structure you can see some small circular shape that are ball-like structure. These ball-like structures are nothing but the tubulins. Okay, this is forming, the single tubulins are forming the whole structure of this microtubule. So what are these tubulins? Here I have shown this tubulins, the structure in two colors. Okay, that is yellow and pink. So what are the structures? So these tubulin structures are nothing but the alpha tubulin and the beta tubulin. These are the monomeric units. So what are the monomeric units? Monomeric is single units that is forming the structure. These are nothing but the alpha and beta tubulin which form the structure. So these monomers, now we will, after knowing the structure, we are going to move forward to the assembly. Okay, how the assembly of microtubule occurs. Okay, in this, the monomeric subunits, as I told, alpha and beta subunits, these are separate, these are monomers. But during the formation of microtubules, this comes together and forms a dimer. Okay, here the dimer position then comes together and form, they get attached one by one. in the dimeric position. Okay, these dimers are arranged, okay, in the straight row, which forms where how many dimers are formed, how many tubulin units are there in a single filament. That is, there are 13 filaments which together it forms a protofilament. Okay, and this 13 protofilaments together forms this microtubule structure. Okay, microtubule structure. So I will show you how is the arrangement of the 13 filaments. See, in this it is formed in a circular manner. So these are arrangement of protofilaments, and below this vertical arrangement of filamentous is done, which together forms this hollow tube-like structure. Here you can see how is the size of the diameter. What is the diameter? Here the diameter is 25 nanometer in size. Okay, so here the diameter is 25 nanometer. Okay, and in this there is a hollow space present in this microtubule. Here you can see that after the formation in the assembly, monomers which are converted into dimers. In dimers there is 13 protofilament formation. But here you can see there is a step where there is nucleotide addition. Okay, why this nucleotide addition is required? The nucleotide which is used in this assembly is GTP and GDP. That is guanine triphosphate is used in this microtubule polymerization. Okay, why they are used? Because there is a binding site at the alpha tubule. Okay, here at this position. Okay, that is in between the interface of alpha and beta tubulin, there is a binding site for guanine. Okay, for guanine at the alpha unit, alpha tubulin. So, and after binding of guanine to the alpha, it becomes active and then it joins the other monomer called as beta subunit. At beta subunit, there is a site for hydrolysis where GTP is converted to GDP. Okay, GTP is converted to GTP. Once again I will tell you, at alpha site there is at N site. Okay, that is N site, N terminal. There is a binding site for guanine. After attachment of guanine, it attaches to the beta tubule. After this formation of, and so it forms a dimer. What happens? Due to attachment of guanine at this beta, alpha and beta interface, what happens? It doesn't give space for the attachment of or for the hydrolysis. Okay, and so it makes the attachment more stronger. So one by one the filaments get attached to it, and then together it forms this structure. So this rigid structure, this attachment is due to the guanine triphosphate. Okay, so this guanine triphosphate attachment helps to polymerize, helps to add the tubulins, the monomers one by one, and increase the size of microtubules. So along with this, as their addition, I would like to tell you, there is this microtubule structure maintains the polarity. It gives some kind of polarity, though it doesn't have any connection with the charge. There is no charge, but this gives the polarity due to assembly. The type of assembly takes place at one end. So the position at where the assembly of this tubulin take place is called as plus end. Okay, where the tubules get attached and cause the polymerization. Means after dimerization there is polymerization forming this protofilaments. This is done at the plus end. Okay, while this is called as minus end. Why? Because the disassembly, okay, disassembly takes place at this end where the polymer units are separated into monomers. Okay, so at plus end there is polymerization, there is addition of tubules. Okay, so this structure is formed, and as we move further, as we go down, there is disassembly system from the minus n. Okay, so when there is increase in the length of microtubule, means the plus end is active and there is continuous attachment, there is continuous assembly of that polymer units, and so here attachment of tubules take place and there is this attachment at this minus n. Why there is this attachment? Why the tubules are separated? This is due to the GDP. GDP means the hydrolysis takes place over there. Okay, after the attachment of GDP at the beta tubulin, the tubules get separated and then the disassembly takes place. So remember, the polymerization takes place at plus end, means that is the assembly, and disassembly, that is depolymerization, takes place at the minus end. But remember, it is not always that 100% there is addition or polymerization at plus end. Also, in minor quantity, there is polymerization at minus n also, but the rate of addition is very less. Okay, so here was all about the structure, the functions, the morphology, the diagrammatic representation of the assembly of the microtubules. So here, with the help of this, we are able to understand what is the microtubules and what are its function in the cytoskeleton. So thank you for watching.